Hello, I'm Joshua Mugabi. I welcome you to Inheritance and Evolution. Learning Outcome D. Examine different forms of allele interaction, autosomal linkage, multiple alleles, codominancy, and incomplete dominancy, including their examples and influence on phenotypic expressions. The learning outcome, we are going to look at A. In pairs, learners search for forms of allele interactions and their influence on phenotypic expression. Make a write-up and present it to the class. We shall start with complete dominancy. Let's emphasize on pea plant flower color. Item 1. A teacher provides pea plants. Purple flower, capital P, are dominants. White flower, small p, are recessive. Students cross heterozygous plants. Capital P, small p, crossed with capital P, small p. Tasks. Construct a Punnett square for the cross. Determine the genotypic and phenotypic ratios. Explain how complete dominance affects phenotypic expression. Construct a Punnett square for the cross. Our Punnett square, we shall have capital P as the gamete, small p, capital P, small p. And when we cross, we get capital P, capital P, capital P, small p, capital P, small p, small p, small p. Determine the genotypic and phenotypic ratios. The ratios. Genotypic ratio 1 capital P to 2 capital P small p to 1 capital P small p. Phenotypic ratio 3 purple to 1 white. Explain how complete dominancy affects phenotypic expression. Explanation. Complete dominancy occurs when the dominant allele masks the effect of the recessive allele in heterozygotes, producing only the dominant phenotype. Let's now look at incomplete dominancy. We shall look at the snapdragon's flower color. Item 2. Red flowers, capital R, white flowers, small r. The heterozygotes, capital R, small r, produce pink flowers. Task. Cross 2 heterozygote pink flowers. Capital R small r crossed with capital R small r. Predict the phenotypic ratio of the offspring. Explain why the heterozygote shows an intermediate phenotype. cross two heterozygous pink flowers, capital R, small r, crossed with capital R, small r. When we cross, the gametes are capital R, small r, capital R, small r, we get capital R, capital R, capital R, small r, capital R, small r, small r, small r. Predict the phenotypic ratio of the offsprings. The phenotypic ratio is 1 red, 2 pink, and 1 white. Explain why the heterozygotes show an intermediate phenotype. Explanation. Incomplete dominance occurs when neither allele is completely dominant. So heterozygotes display a blended phenotype. Let's now go to codominancy. We shall look at human blood groups. We have allele immunoglobulin A, immunoglobulin B, and I. IA and IB are codominant. I is recessive. Task. Cross a parent with blood type IAI with one IBI. Determine the possible blood types of offspring. Explain how codominancy affects phenotypic expression. Cross a parent with blood type IAI with one IBI. The Punnett square will give us gametes IA, I, IBI. Shall get IAIB, IBI, IAI, and II. Determine possible blood types of offsprings. The possible offspring blood types: IAIB, that is blood group AB, IAI, that is blood group A, IBI, that is blood group B, II, that is blood group O. Explain how codominance affects phenotypic expression. Explanation. Codominance occurs when both alleles are fully expressed in the heterozygote, resulting in a phenotype that displays both traits simultaneously. For example, AB blood type. Let's now look at multiple alleles. We shall look at the rabbit coat color. Item 4. Alleles C, full color. CH, chinchilla. CD, dilute. And C, small, albino. Explain how multiple alleles increase phenotypic variety. Give an example of a cross showing more than 2 possible phenotypes. Explain how multiple alleles increase phenotypic variety. Explanation. Multiple alleles means more than 2 forms of a gene exist in a population. Give an example of a cross showing more than 2 possible phenotypes. Example, a cross between capital C, CD, capital C, CH. The possible phenotypes will be one with full color, we shall have the chinchilla, and we shall have the dilute. Explanation. More alleles increase the number of phenotypic combinations possible. Let's now look at epistasis. We shall look at coat color in mice. Item 5. One gene, capital B, smaller b, determines black, capital B, or brown, smaller b, pigments. Another gene, capital C, smaller c, controls pigment deposition. If you have capital C or capital C small c, pigment is deposited. If you have small c, no pigment, that means an albino. Task. Cross 2 heterozygous mice, capital B, smaller b, capital C, smaller c, crossed with capital B, smaller b, capital C, small c. Predict the phenotypes of the offsprings. Explain how epistasis modifies expected ratios. Cross 2 heterozygous mice, capital B, smaller b, capital C, smaller c, crossed with capital B, smaller b, capital C, smaller c. Gametes, we shall have capital B, capital C, capital B, small c, smaller b, capital C, smaller b, small c. Predict the phenotypes of the offsprings. Punnett square analysis shows phenotypes: black 9 out of 16, brown 3 out of 16, albino 4 out of 16. Explain how epistasis modifies expected ratios. Explanation. Epistasis occurs when one gene masks the expression of another gene. In this case, the small c genotype prevents any pigment overriding the capital B, smaller b gene. Combined activity class experiment. We are going to use bean seeds to demonstrate allele interactions. Black seeds, capital B, they are dominant to white, small b. Smooth texture, capital S, is dominant to wrinkled, small s. Students simulate complete dominance, capital B crossed with small b. Codominance, use red and white beads mixed to show both colors. Epistasis, assign a gene that prevents color production. Task, arrange gametes for each scenario. Record phenotypes of offspring. Discuss how different forms of allele interactions affect traits. Arrange gametes for each scenario. You can try this one out. Pause the video and try it out. Record phenotypes of offsprings. Complete dominancy. The heterozygotes show dominant traits. Codominancy. Heterozygotes show both traits simultaneously. Epistasis. Masking gene alters expected phenotype ratios. Discuss how different forms of allele interactions affect traits. Allele interaction dictate phenotypic diversity. Dominance relationship, codominance, incomplete dominance, multiple allele, and epistasis all explain why genotypes may not directly predict phenotypes. Thank you very much for participating in Inheritance and Evolution. Learning Outcome D. We have been examining different forms of allele interaction, autosomal linkage, multiple alleles, codominance, and incomplete dominance, including their examples and influence on phenotypic expression. We have been looking at learning activity A. In pairs, learners search for forms of allele interactions and their influence on phenotypic expression. They make a write-up and present it to the class. If this interaction has been useful to you, visit me at Mugabi Joshua Biology on YouTube.